Next, we're going to look at the structure of DNA. Cells contain large quantities of DNA and protein. Scientists were trying to discover which of these types of molecules acted as the genetic material. Scientists knew the existence of the 4 types of nucleotides. Chargaff examined the DNA of a variety of different species of organisms and found that the amount of adenine always equals the amount of thymine, or A equals T, and the amount of cytosine always equals the amount of guanine, or C equals G. All nucleotides contain a phosphate, a 5-carbon sugar, deoxyribose sugar in DNA, ribose sugar in RNA, and a nitrogen-containing base. This diagram shows the phosphate group, ribose sugar, and one of the nitrogenous bases. This diagram shows the 4 nitrogenous bases found in DNA. Cytosine and thymine are pyrimidines, which means they have a single ring structure. Guanine and adenine are purines, which mean they have a double ring structure. In the DNA molecule, one of the smaller nitrogenous bases is paired to the other. Therefore, A goes to T, and C goes to G. Rosalind Franklin studied the X-ray diffraction pattern of DNA. Her data showed that DNA to be a helix with portions that repeat over and over again. James Watson and Francis Crick developed a model of DNA. Their model holds true today with few changes. They won the Nobel Prize Science in 1953. The structure of DNA is that of a double helix. The 2 strands are twisted together. The deoxyribose sugar and phosphate molecules are bonded together and form the sides of this twisted ladder, and the nitrogenous bases make up the rungs. Remember that adenine always bonds with thymine and guanine with cytosine. This diagram shows you the sides as well as the rungs of the ladder. Before mitosis can begin, remember that DNA must be replicated. DNA replication is the process of copying DNA before cell division occurs. In order for this process to be accomplished, the 2 strands need to separate. Each of the original strand serves for a template for the new strand. DNA replication is semi-conservative. Each new DNA molecule is made of 1 parent strand and 1 new strand. This diagram shows how the DNA molecule unzips and the 2 new strands start to form. RNA molecules are the worker molecules of the cell. Ribonucleic acid consists of the sugar ribose, and instead of having the nitrogenous base thymine, this is replaced by uracil. RNA molecules are single-stranded. There are 3 major types: messenger RNA, transfer RNA, and ribosomal RNA. This diagram shows the difference between ribose sugar and deoxyribose sugar. This is a very important slide for you to learn. Let's look at the similarities between DNA and RNA. They're both chemically classified as nucleic acids. They both consist of nucleotides. They both have sugar phosphate backbones, and they both have 4 different types of nitrogenous bases. Now let's look at the differences. DNA is found only in the nucleus. RNA is manufactured in the nucleus, but leaves the nucleus and enters the cytoplasm. DNA functions as a genetic material inside a cell. RNA molecules are helpers to DNA. The sugar in DNA is deoxyribose, while the sugar in RNA is ribose. The 4 nitrogenous bases found in DNA are adenine, thymine, cytosine, and guanine. In RNA, the 4 nitrogenous bases are adenine, uracil, cytosine, and guanine. All DNA molecules are double-stranded, while RNA molecules are single-stranded. DNA can be copied or transcribed to give rise to the 3 types of RNA: the messenger RNA, transfer RNA, and ribosomal RNA. Messenger RNA is translated, and this generates the production of proteins. Let's look at the three types of RNA and how they function in the production of proteins. Messenger RNA, or mRNA, is produced in the nucleus from a DNA template. Messenger RNA is a copy of the gene for a particular protein that needs to be manufactured. This carries this genetic message to the ribosomes, the organelle inside the cytoplasm where protein synthesis occurs. Proteins are made of changed amino acids. It's a function of transfer RNA to transfer the correct amino acid to the growing amino acid chain. Ribosomal RNA is also produced in the nucleus and is an integral part of the ribosome. Let's look at gene expression. A gene is expressed whenever the particular type of protein that is coded for is produced. DNA provides a blueprint to synthesize these proteins. The information flows from DNA to RNA and then RNA to the production of the protein. Some individuals have identical DNA, such as identical twins or monozygotic twins. This is where one fertilized egg or zygote splits in half. Some twins are conjoined. This is where the separation or splitting of the zygote is not complete. Fraternal twins are dizygotic. This results from two separate embryos. Pairs are genetically related to each other as other brothers and sisters. The central dogma describes the flow of information from DNA that encodes for RNA and RNA which then encodes for protein. This is how protein synthesis occurs. Protein synthesis occurs in two main stages. Transcription, which occurs in the nucleus, is where DNA serves as a template to manufacture the messenger RNA. Inside the cytoplasm, translation occurs. This is where the messenger RNA directs the sequence of amino acids to form the new protein. The ribosomal RNA and transfer RNA assist. The ribosome is a protein synthesis machinery. It consists of a large and small subunit. On this diagram, you can see the growing amino acid chain. The DNA nucleotides code for the specific amino acids and specify their correct placement. Let's stop here for a minute. There are 4 nucleotides: adenine, thymine, cytosine, and guanine. There are 12 naturally occurring amino acids. So, if each nucleotide was read singularly, you would only be able to code for amino acid. Therefore, the nucleotides are read in sets of 3, or triplets. Each 3 nucleotides in a sequence of DNA code for 1 amino acid. This is referred to as a codon. 3 nucleotide sequence in messenger RNA. Each codon codes for a single amino acid. If you take 4 letters and select 3, there are 64 possible combinations. So therefore, each amino acid has many different codons that code for it. There are also some stop as well as start codons. Remember that when DNA is transcribed into messenger RNA, thymine is replaced by uracil. This shows a messenger RNA dictionary where you can look at any codon set of 3 nucleotides and determine what amino acid it represents. Okay, maybe phenylalanine or leucine or isoleucine or proline. You do not have to worry about what abbreviation stands for which amino acids, but you do need to be able to read the messenger RNA dictionary. Let's look at the following problem. Given the following DNA coding strand sequence, what would be the messenger RNA? Remember, in DNA, A would go to T, but in messenger RNA, thymine is replaced by uracil. So we would have U, A, U. If we look at the DNA dictionary and find U, A, U, the answer would be tyrosine. Let's look at the next example. I'm going to write this one above it. G goes to C, right? C goes to G, G goes to C. So our next amino acid in our growing protein strand would be C, G, C, arginine. Let's look at our third codon. T goes to A. Remember messenger RNA, A goes to U, and C goes to G. So A, U, G, or methionine. Let's look at the next problem. Select the correct amino acid for AUA. We have AUA, isoleucine. We talked about genetic mutations in our previous unit. Genetic mutations occur when there's changes in the sequence of bases in a gene. This can be caused by replication error. However, this is fairly rare due to proofreading. There, however, are mutagens. These are substances that induce mutations in DNA. These can be caused by environmental influences, such as radiation or asbestos. There are chemicals, such as nitrates, that can cause genetic mutations. Normally, there are repair mechanisms or repair enzymes that correct these changes. However, sometimes the genes coding for these repair enzymes themselves become mutated. Some types of mutations are called silent. They may go undetected and not have any observable effect. For example, if there is mutation in our DNA sequence that caused the messenger RNA, instead of being CCU to be CCC, in both cases the amino acid proline is selected, so there wouldn't be any observable effect of this change. Sometimes, however, a change in one DNA nucleotide can cause a rather severe genetic disorder. When one single DNA nucleotide is mutated or changed, this is called a point mutation. An example of this is sickle cell disease. In sickle cell disease, the amino acid valine replaces glutamic acid. This changes the solubility of the hemoglobin molecule, and under instances of stress or increased oxygen demand, the molecule comes out of solution, causing the red blood cell to sickle. More severe is a frame shift mutation. This is where a nucleotide is added or deleted. Look at the sentence, "The cat ate the rat." Okay, I'm kind of using some imaginary letters, but just think of the letter C as being a nucleotide that's removed. Now the sentence or the codons read, "The atta tat her at." So it's going to affect every amino acid down the line after this mutation has occurred. Polymerase chain reaction, or PCR, is used in DNA fingerprinting. What it does is it amplifies specific DNA sequences. This utilizes the enzyme DNA polymerase, which is used in the synthesis or replication of DNA. It's extracted for Thermus aquaticus, a water bacteria that can tolerate high temperature. DNA fingerprinting makes use of non-coding DNA segments. These segments are found in between our coding genes. These non-coding segments have almost hiccups or repeating sequences, and individuals differ in how many repeats that they have. PCR is used to increase sample size. And then restriction enzymes, which are isolated from bacteria, cuts the DNA into fragments. The fragments that are generated vary in length, and each individual has their own unique set of fragments. Electrophoresis is a process that separates samples by size. DNA molecule has a negative charge. An electrical current is run through a gel loaded with the DNA sample. The DNA fragments will migrate toward the positive pole. Longer DNA strands or fragments are larger, and they will migrate less on the gel. This diagram simulates a DNA fingerprint. Crime scene evidence is obtained. The DNA goes through the PCR process, which increases its size, and then the restriction enzymes generate fragments, and this shows the two fragments from this DNA sample. Here we have DNA samples from suspect A and suspect B. You can see that suspect B has the same exact fragments as isolated from the crime scene. This means that his DNA is the same DNA that was collected. This slide shows evidence for a rape case. Here are the fragments that are isolated from the victim. suspect 1, suspect 2. Here we have the male vaginal extract. This is the male DNA that was isolated from the female's vagina. As you can see, this matches the fragments that were isolated from the DNA sample from suspect 2. During the Russian Revolution, Tsar Nicholas and his family, his wife and 5 children, and 2 servants were executed. 9 bodies were found in a shallow remote area in 1991. This grave contains 6 adults and 3 children. The bodies had bullet and stab wounds and gold dental work, and they were found near the area of the exile. DNA fingerprints confirmed that these were indeed the royals and their servants. However, there were 2 bodies that were missing. This slide shows the remains of these bodies that were found. The 2 smallest children, Anastasia and Alexis, were missing from the grave. There are many individuals that claim to be the surviving children of the Russian Tsar. 2 additional bodies were found, and DNA fingerprinting disproved these individuals. I really suggest that you watch this video, Mystery of the Room, in its entirety. It's very interesting for its biological and historical significance.